Hello, my name is Sonia Fronten, and today I'll be presenting on the paper, Timing of Tissue-Specific Cell Division Requires a Differential Onset of Zygotic Transcription During Metazoan Embryogenesis. The paper was written by Ming-King Wong et al. And was published in the Journal of Biological Chemistry in June 2016. So to give a little bit of context, cell division usually varies from organism to organism. For the most part, the process involves an animal cell where the mother cell divides into two identical daughter cells. This process in itself is usually synchronized and very well timed, and the length of this cell division cycle is important in determining how quickly an organism can multiply. In this study, we're using the complete opposite and actually looking at the study of asynchronized cell division in these cells with the use of a metazoan organism. And the role of this organism in development is poorly understood, which made it a good study. Organisms from these cells involve significant differences in gene expression and genome modification when compared to cells that undergo synchronized division. With no synchronization in their division, these cells typically see a delay in cell division in two endoderm progenitor cells, also known as E2, that are responsible for endoderm germ development. Now, endoderm germ levels are known for your internal layer development, which include lung cells, thyroid cells, pancreatic cells, and the GI tract, among many others. The goal of this study was to study the timing and quantifying rapid cell division in non-synchronized cell division and the role of genetics and transcription factors of a zygotic nature, specifically the endoderm progenitor cells, during this process. The hypothesis that I gathered from this paper was that cell zygotic factors and subsequent activation is required to coordinate cell fate differentiation, cell division timing, and cell migration to ensure proper organism development. The population that was used was from the metazoan family, which I will give you a little bit more information about. It is from the kingdom Animalia, which is also known as metazoa. Typically comprises of multicellular organisms which perform incorporation with many other similar cells within an internal body. It undergoes development from an embryo stage with three tissue germ layers, which include your ectoderm, mesoderm, and endoderm, and is typically classified under 36 types, which I will not all go through. But the organisms can include humans, birds, snakes, reptiles, insects, snails, squid, jellyfish, coral, and in our study, worms. The population used for this study is C. elegans, and it was maintained on an NGM plated E. coli OP50 at room temperature. These worms thrive well on these plates with E. coli that they feed on, and they were actually chosen as the organism of study because their poorly understood coordination through development due to the invariant development and widespread divisions that are not synchronized. They are also transparent, so they're easy to study and see under a microscope. They also are hermaphrodite or male, so the hermaphrodites can actually produce asexually or can mate with a male and produce offspring. And they have a short lifespan as well as a lot of offspring when they do mate and produce offspring. So the design and methods of this study was first to unravel the genetic code over E2 division timing. 822 essential and conserved genes were actually knocked out using an RNAi, and then the sense cycle lengths using in toto imaging of the organism was done. The same procedure was done for the wild type organism where no genes were knocked out for a comparison. The manual measurement of the cell cycle length using fluorescent imaging, differential interference imaging, was also used to manually trace the E2 cells from birth to division and have a better idea of the cell cycle timing during this study. Statistical analysis was performed on the cell cycle length using Starry Night software and deposited in a Phoenix database. To identify any deviations in cell division timing from the wild-type cells, the D'Agostino Chi-square test was performed to evaluate the distribution of the E2 factors in wild-type cells as compared to the cells that were used with the RNAi knockout procedure. Cells that produce a significant increase or decrease in the E2 cell cycle due to deviated genes were pooled together to build a matrix of cell cycle lengths. Only cells with these changes in at least 2 embryos were further analyzed. If they only were seen in 1 embryo, those were not studied further. So a couple models and figures that were seen in this paper. As you can see in figure A, the first rise and peaks were for the wild type, while the second structure is for your knockout organism. And as you can see on the y-axis, the development time in minutes. They were able to match up specific points in time in development and see that there was a significant decrease in the knockout specimen in cell division timing when comparing your wild type to the knockout organism. Figure B shows the ontology of the genes with a significant reduction in the E2 cell cycle length after using the knockout organism and going through the methodology to figure out the timing of the cell division process and comparing it to your wild type and seeing that there was actually a significant reduction in the amount of time it took for the cell division to happen. Another study here shows your wild type in purple. On the y-axis is your cell cycle timing in minutes, and as you can see, the cell division control wild type took the most time in cell division. All the other columns represent other genetic strains that were all altered by RNAi and used by knockout genes, etc. And in these columns you can see that the cell division was much shorter timed in each one of these cells that were altered using RNAi. And the comparison of the wild type and the deviated transcription factor genes resulted in the E2 cell cycle length, and all the deviated transcription factors were altered, and the deviated genes are shown here. There are all the bars except for the dark purple, which is labeled wild type. So the conclusion here, based on the experimental data, the hypothesis was concluded to be true. Transcription factor E2 was significantly seen to reduce cell cycle length. This reduction in cell cycle is often associated with migration defect and gastrulation failure. The results suggest that a tissue-specific transcriptional activation is required to coordinate fate differentiation, cell division timing, and cell migration to ensure proper development. Evidence also showed that the tissue-specific regulation of the cell cycle length requires a differential onset of gene transcription. Better understanding of these transcription pathways are needed, and that was specifically honed on and mentioned throughout this article. So in reviewing this article, there were also additional opportunities that the authors thought would be beneficial for further studies, and this included the investigation of the exact relationship between the initiation of zygotic transcription and gap checkpoint introduction during the mitosis process, whether an increased E2 cell cycle length contributes to the activation of zygotic transcription in C. elegans remains to be determined, whereas in your normal cell cycle length there will be normal cell development. So if it is that there is an activation of zygotic transcription based on an increased cell length to bring that cell length back to something more normal, would that be something that can be conducive to normal cell development. Thank you for your time, and I look forward to your feedback.